So I just want to remind everyone that the CME code for today's lecture is 37672, and for people who are...
Is that correct?
Yeah.
5767?
No, 37672. And for those of you who...
That's not correct. I think it's 57.
Oh, it's 57672. Oh, my. Okay, yes, sorry about that. Okay. So I also want to let people know who are watching remotely that if you have any questions for the speaker, you can enter them using the Send Live Feedback key that you should have on your monitor. Okay, so on the behalf of the Membrane and Organelle Organelle Dynamics Special Interest Group, it is a great pleasure to introduce today's speaker, Dr. Tom Rapoport, who is currently the Don W. Fawcett Professor of Cell Biology and an HHMI investigator at Harvard Medical School. Tom is back by popular demand for his third Wednesday afternoon lecture, which at least ties, if not sets the record for the most Wednesday afternoon lectures given by an individual speaker since the series began 30 years ago. As many of you know, Tom's remarkable contributions to the field of cell biology have been paralleled by his remarkable life story, both of which are described in an autobiography that he published last year in the Annual Review of Biochemistry entitled A Life of Translocations. To summarize, Tom was born in Cincinnati, but then moved with his family at a young age to East Germany, where he received his Ph.D. and led a research group at the Institute of Molecular Biology in East Berlin. He was given a budget of only about $1,000 a year, but he was one of a very small number of East Germans who was allowed to travel to the West, where he gave lectures and met with some of the rising stars in the protein trafficking field, who generously donated some basic supplies, such as Eppendorf tubes and pipette tips. And it was during one of his adventures in the West that I first met him. Despite facing many obstacles, Tom's group was still able to publish a seminal paper in Nature in which they identified the subunit of the signal recognition particle, or SRP, that binds to signal peptides. After the reunification of Germany, Tom's lab continued to do groundbreaking work and showed that the eukaryotic Sec61 complex, which is closely related to the Sec-Y complex in bacteria, forms the channel through which proteins are translocated into the lumen of the endoplasmic reticulum, or ER. Unfortunately, Tom once again faced barriers because he was seen as having profited from what became a malign political system. And so in 1995, he decided to move to the cell biology department at Harvard, where he has remained ever since. So at Harvard, Tom's work really took off, and over the years, his group has made an incredible number of important discoveries. And just to mention a few, they solved the crystal structure of the Sec-Y complex, which was the first structure of any protein conducting channel to be published. And they used the structure to explain how the complex can catalyze the transport of proteins both across and into a cell membrane. They also identified and characterized the key factors that promote the retrotranslocation of misfolded proteins from the ER back into the cytoplasm. They elucidated the mechanism of ER-associated protein degradation, or ERAD, and they identified the proteins that give the ER its characteristic reticular structure. In the last few years, Tom's group has tackled one of the most daunting problems in cell biology, namely how folded proteins are transported into peroxisomes, and showed in a paradigm-shifting study that the import reaction is remarkably similar to nuclear import. Not surprisingly, Tom has been the recipient of numerous awards, including the Otto Warburg Medal of the German Society for Biochemistry and Molecular Biology, the Max-Delbrueck Medal of the Max-Delbrueck Center for Molecular Medicine, the Sir Hans Krebs Medal, and the Schleiden Medal, and he was elected to the National Academy of Sciences in 2005. Personally, I have always been amazed by Tom's ability to find elegant ways to address challenging problems in cell biology using a broad range of methods. His skill in mentoring trainees, many of whom have gone on to highly successful careers, and his modesty. Just to quote from his credo that appeared in his recent autobiography, "For me, the ideal project begins with a complex phenomenon in an intact cell and leads all the way to a defined system in which the process is recapitulated with purified molecules and understood at a structural and mechanistic level. There is nothing that can compare with the excitement of a real insight and discovery. For me, a nice experiment, however small, can make my day." So as you can see, well, I guess you can't see, the title of Tom's talk is How Proteins Cross Membranes. Tom, thank you very much for translocating once again from Boston to the NIH on such a cold and energetically unfavorable day, and we're really looking forward to hearing about your recent work.
Okay, well, in the remaining time, well, thank you, Harris, for this very nice introduction. I'm grateful that so many people showed up, even though the weather is really not very cooperative here. It's very cold. This has been a difficult week, I think, in many respects, not because of the temperature so much, but more because of the political situation. And, I think we're all pretty upset with what's going on. And I'm sure you're particularly concerned about what's going on with NIH. I just heard that there's a travel ban that was announced today. You're not alone. I think Harvard is on the shit list as well. On the positive side, the football team is doing pretty well. Much better than the Patriots, who really stink. Okay, so what I'm going to talk about is how proteins cross membranes, and this is more or less, this is a funny pointer here. It's kind of jumping around. I will try to give you an overview of some things, but I will mostly concentrate on peroxisomal protein import. But let me start out by making a few general comments. So as you all know, most proteins are synthesized in the cytosol on cytosolic ribosomes, but then eventually they end up in different sites of the cell, for example, different organelles, and often they have to traverse a membrane in order to get to their final localization, and this means that they have to cross a membrane, which is energetically difficult. So membranes are impermeable for even small molecules, let alone macromolecules. So what I will tell you about is different solutions to this problem. And the one solution that we've known about for many years is translocation through an aqueous channel. It's obvious that if you have a hole in the membrane with an aqueous interior, that will break down the energy barrier dramatically and allow a polypeptide chain to go across. And this system has been established for the Sec61 channel in the endoplasmic reticulum membrane and the analogous Sec-Y channel in the plasma membrane of prokaryotes. Just to remind you, there's a substantial amount of proteins that are initially translocated across the R membrane. Some proteins make it all the way across the membrane and become soluble in the lumen. Others are integrated into the membrane. And in both cases, they use the Sec61 channel for translocation. And then there's a vesicular transport phase when vesicles bud off from the R, take the proteins through the Golgi all the way to the plasma membrane. Eventually, these vesicles fuse with the plasma membrane, deliver the content to the outside, so these become secretory proteins, and the membrane proteins become membrane proteins of the plasma membrane. And of course, proteins can also stay in different locations along the secretory pathway or be diverted to the endosomal system. But the important point is that this is the initial stage here, the translocation phase, where the decision is made whether proteins are entering the secretory pathway. Now, secretion in bacteria and archaea occurs through the plasma membrane, through the Sec-Y channel. And the relationship between Sec-Y channel and Sec61 channel is easily explained by evolution. This is a primordial single-cell organism, probably by some kind of bacterium. And then you got invagination of the plasma membrane, eventually pinched off became an intracellular membrane. And so the Seg-Y channel now is translocating, or Seg-61 channel is called, into the lumen of the ER, or integrating into the membrane of the ER. Obviously, you need also vesicular transport to move the protein from the internal membrane back to the plasma membrane. So in 2004, we were able to get the first structure of a protein-conducting channel. That was a Seg-Y channel from an archaeobacterium. There were 3 postdocs involved, Bert Vandenberg, Bill Clements, who are here, gave this lecture just recently, and Ian Collinson, and this was a collaboration with Steve Harrison, a colleague of mine. So what you see here in the top is a view from the cytosol. You're looking down onto the membrane, and you can appreciate that the large subunit, the alpha subunit, consists of 2 halves, transmembrane segments 1 through 5 and 6 through 10, shown here in blue and red. And the protein has and they're linked on the extracellular side by a hinge, and they can open on this side, so like my fingertips do here. So it's like a clamshell that can open sideways. We call this the lateral gate. And then in a side view, you can see from this space-filling model that there is a cytosolic cavity and an extracellular cavity. The cytosolic is empty, but the extracellular is filled with a little helix. It's called the plug. And the constriction in the middle is formed by 6 amino acids, hydrophobic, that project their side chains inwards. So this channel is opened when a translocating polypeptide arrives. So this is, for example, the secretory protein. The signal sequence is shown here in red. And then the polypeptide chain inserts as a loop into the membrane. The hydrophobic sequence moves sideways out through the lateral gate into the lipid, and the plug moves out of the way, and then the other part of the hairpin moves through the aqueous channel into the lumen. Eventually you get single sequence cleavage in the case of secretory protein, and the protein ends up on the luminal side. This also works for membrane proteins. In this case, the hydrophobic sequence is not cleaved. So this shows the actual crystal structure that supports this model. Here's a secretory protein with a single sequence shown here, sitting outside the lateral gate at the interface between the channel and the lipid. And if you turn this structure around, you can see that the polypeptide chain shown here in blue, this is not fantasy, this is actually the actual structure, moves from the cytosol through the pore ring, shown here in red, through the extracellular space. The plug is moved out of the way. And this is work from Long Li and Unyoung Park, 2 postdocs, former postdocs in the lab. So in the case of secretory proteins, they're moved completely across the membrane through the channel. In the case of membrane proteins, they're only partially translocated, meaning some segments are translocated, others stay behind in the cytosol. And so the important point here is that the polypeptide chains move through a channel that has an aqueous interior. There's water here, there's water down there. The membrane barrier is confined to a single layer of amino acids. And this obviously reduces the energy barrier for moving the polypeptide chain through the membrane. And the other point is that translocation is through a gated channel that only opens when the substrate arrives. And finally, I want to make the point here that the barrier for small molecules is maintained during translocation of the macromolecule, which is not a trivial thing if you think about it. So in the idle state, the plug is right underneath the pore ring, and there's no free flow of ions. And when the plug is out of the way, the translocating polypeptide chain sits in the pore, and the pore ring residues sit around the polypeptide chain like a gasket and prevent, again, the free flow of ions. And when the polypeptide chain is moved into the lumen or sideways, in the case of membrane proteins, the plug can return and close the channel. So at no time during translocation or in the idle state is there a free flow of ions possible, and you maintain the small molecule barriers imbalance. So for a long time, the whole field, including us, believed that this is the only way you can move a polypeptide chain through a membrane. But then it turns out this is not the case, and we noticed this by studying ERAD, ER-associated protein degradation. So let me give you a little bit of background. I will talk more tomorrow about ERAD, if you want to come to that lecture. So again, we're first talking about forward translocation, which is what I just talked about, the Sec61 channel allowing polypeptide chain to move all the way into the lumen or to be integrated into the membrane. In both cases, proteins undergo quality control such that only properly folded proteins move on along the secretory pathway or become resident in the ER. If you don't reach the native folded state, these proteins are moved back into the cytosol through some conduit, and then we call this retrotranslocation because it's the opposite direction of how proteins came in the first place. Then usually you get polyubiquitination, and finally the proteins are degraded by the proteasome. And this is the process that we refer to as ER-associated protein degradation, or for short, ERAD. Now we're particularly interested in ERAD-L, which is the degradation of misfolded luminal proteins. The reason is that, and this is the most challenging pathway, because you're starting in the lumen as a soluble protein, and then you move it all the way into the cytosol for degradation. So how do you get it across the membrane? Turns out for this you need a protein complex that we call the HERT1 complex. It contains the HERT1 protein, which is a ubiquitin ligase, a ring finger ligase, and the DIR1 protein, which is a rhomboid-like protein. These two are multi-spanning membrane proteins. There's a linker protein called USA1 that links HERT1 and DIR1. And then there are two proteins with large luminal domains, YOS9 and HERT3. So we determined a cryo-EM structure which told us how this might work. So here's a HERT1 protein, the DIR1 protein, the linker protein, USA1 protein. YOS9, the luminal protein, has two domains, and here's the HERT3 protein. And so it turns out that the proteins that are actually responsible for moving polypeptide chains through the membrane are the two multi-spanning membrane protein, HERT1 and DIR1. And these two proteins have lateral gates that face each other in the plane of the membrane, as shown here. You're looking from the ER lumen onto the membrane. And it turns out that there is no actual channel. Instead, you have a thin membrane area at the interface between the two proteins, the HERT1 proteins and the DIR1 protein. This is shown here in this cut through a space filling model. So you see that there's a cavity on the cytosolic side, and there's also a cavity on the luminal side. And together, they thin the membrane to about 1/2 of the thickness of a normal bilayer. And this is also confirmed by molecular dynamics simulation that we did together with a group of Gerhard Humboldt. So the point here is that this thinning of the membrane would, again, reduce the barrier to move from polypeptide chain through the membrane. Now, we don't have a structure of the substrate with the HERT1 complex. However, we have substantial cross-linking experiments. And so from these experiments, we know that the substrate, the misfolded polypeptide, inserts as a loop with one part of the hairpin inserted into HERT1 through the lateral gate. The other part of the hairpin goes through the lateral gate into DIR1, and the tip of the loop moves through the thin membrane area to the other side of the membrane. And then presumably the polypeptide chain can slide back and forth until a suitable lysine residue arrives on the cytosolic site, which then can be polyubiquitinated by the HERT1 ubiquitin ligase. So these experiments led to a new paradigm. Where is the new paradigm here? Oh, here it is. So the new paradigm is that proteins are translocated to a locally thinned membrane area. So the reason the membrane is thinned is because both HERT1 and DIR1 have relatively short transmembrane segments in this area, and so this allows water to come in on either side. So we believe that this translocation to a locally thin membrane is actually a pretty general mechanism. I will mention tomorrow other processes in which this mechanism seems to apply. And we think that the thin membrane region could still maintain the barrier for small molecules. So now I'm going to switch to peroxisomal protein import, because by studying peroxisomal protein import, we discovered two novel ways of how proteins can move through a membrane. So let me first introduce the peroxisomes, because they're not as famous as the endoplasmic reticulum or mitochondria or something like that. So these are membrane-bounded organelles. They are found in most eukaryotic cells. They house many metabolic reactions, including those that are involved in the degradation of long and branched fatty acids and in the reduction of hydrogen peroxide. And they are medically important because they're peroxisomal disorders. The most severe one is Zellweger syndrome, where the children die after about 2 years. So, without exception, all proteins are made in the cytosol and then imported into the lumen or matrix of the organelle post-translationally. So shown here, so you first complete the polypeptide chain in the cytosol, and then you move them across the membrane into the matrix. We know many of the import components from genetic screens. They're called PEX genes, PEX and a number. Makes it very difficult for people outside the field to understand which protein is which. So hopefully you don't get too much confused with the numbers. Until recently, however, the mechanism of protein import was really poorly understood. And one of the most mysterious aspects, as Harris was pointing out, is that folded and even oligomeric proteins can pass the peroxisomal membrane. And this is very different from what we know about translocation across ER, mitochondrial membrane, or chloroplast membranes, in which proteins are unfolded during membrane passage. So most proteins that are imported into the matrix have a C-terminal signal that that is called in the field PTS1, for peroxisomal targeting sequence 1. It's a very simple signal: serine, lysine, leucine, SKL. It's at the C-terminus. And this is recognized by a soluble receptor called PEX5. PEX5 has a folded domain at the C-terminus. It's called the TPR domain. It's shown here folded, and it binds the SKL sequence here in this crystal structure. PEX5 also has a very long unstructured N-terminus, and in the N-terminus are several of these aromatic motifs that read W, XXX, and then F or Y. There are different numbers in different species. Yeast has only 3, and higher organisms there are more. And then there's a cysteine that's conserved, and there's near the N-terminus, and I'll come back to this in a minute. So these cargoes are recognized by PEX5 through the SKL and TPR domain in the cytosol, and then this complex moves all the way across the membrane into the lumen. This translocation occurs through the docking complex that contains 2 conserved subunits, PEX13 and 14. So I will first talk about how this works for the PTS1 cargo, but obviously if there's PTS1, there's PTS2 as well. So in the later talk, I will mention how proteins of the PTS2 class are imported. So it turns out that import of PTS1 cargo works like nuclear transport. So just to give you a little bit of, remind you of how this works. So nuclear pores are formed from nucleoporins, and several nucleoporins have phenylalanine glycine-rich FG repeats that form a meshwork inside the pores. So my former student, Dirk Ehrlich, came up with this selective phase model. So the FG repeats are inside the nuclear pore, and they interact with one another and form a meshwork with a limited pore size, such that small molecules can move through, but large molecules are excluded. The nuclear transport receptors, NTRs, are big, but they can locally break this meshwork by interacting on the surface with the FG repeats. And so they can diffuse through the meshwork and take folded cargo with them. So we noticed that PEX13, one of these docking protein components, has a tyrosine glycine-rich YG domain, which obviously, except for the Y, and instead of the Y you have an F. Otherwise, it looks pretty similar. So here you see the YG domain from civil species, and you can see that there is no sequence similarity between the different species, but all have quite a number of these tyrosine residues. In some cases, you have phenylalanine residues shown here in green. And then these aromatic residues are separated by spaces that are rich in glycines and serines. And that's why we call it the YG domain. The YG domain in PEX13 is preceded by an unstructured region and followed by an amphipathic helix. And in most species, you have a transmembrane segment and an SH3 domain as well. However, these two domains here at the C-terminus are not universally found in all species. Plants and algae lack these two domains, so only this region here is really essential. And this is also, if we make mutations in these two domains, transfer key import is actually abolished. So if PEX13 forms a nuclear pore-like structure, it would have to form oligomers. And so in order to test this, we used yeast and we introduced into the YG domain cysteines at different positions here. Then we isolate the membranes and add an oxidant to induce disulfide bridge formation between the cysteines. So let's look first at cysteine 104. If you add increasing concentration of the oxidant, you convert the monomer into a dimer. If you add detergent, this dimer formation is disrupted. And also, if you reduce the disulfide bridge by adding DTT, there's no dimers. And this works for 131 as well, so you get in both cases efficient dimer formation. So we can also introduce both cysteines at the same time, and now you can see that you form a ladder. And again, if you add DTT, all these species collapse to the monomer. So we can simply count here how many bands you would see here to estimate the number of molecules that form the oligomer. Our estimate is 12. I have to say, though, that this doesn't mean that it's a homogeneous species of 12 copies. It could be a range of molecules, so it could be heterogeneous in composition. Yeah, so probably 12. So what I'm not going to show you is that the YG domains form a meshwork that excludes large but not small molecules. We showed this by accessibility of luminous cysteines to modification reagents. And also what I'm not going to show you is that PEX13 sits in both orientations in the membrane. So there's about 50% of the molecules sit in this orientation with the C-terminus in the lumen and the N-terminus on the other side, and 50% have the opposite orientation. This is a very unusual property of a membrane proteins. Most membrane proteins sit in the membrane in only one orientation, but in this case, we have a random orientation of the protein in the membrane. And this is important because this allows the YG domain to be suspended in the middle of the membrane, which wouldn't be possible if it was only sitting in one orientation. We believe that the walls of the channel are formed from the amphipathic helix that I mentioned earlier that immediately follows the YG domain. For example, if we give AlphaFold 12 copies of the amphipathic helix, it will arrange them in a ring structure in alternating orientation. So this molecule has the C-terminus here, the next one has it on the other side, and so on. And the interesting thing is that the outside of this arrangement is hydrophobic, as you would expect, because this faces the lipid bilayer, and the inside is hydrophilic and would accommodate the YG meshwork. So the next question is whether the YG meshwork actually allows PEX5 to partition into it, and actually also does the YG domain even form a meshwork at a real phase. And this has obviously been shown before for the FG repeats for nuclear transport receptors. So what we did is we expressed N-terminal fragments of PEX13 in bacteria, either the YG domain alone or the YG domain plus the preceding unstructured region, and then purified the proteins under denaturing conditions. Then we can put the solution into a silicone tubing and wait until the protein gelates. And then we push it out of the silicone tubing, and what you see is a hydrogel. And so if we mutate all the tyrosines into serines, the protein remains fluid. And so this makes us believe that the YY interactions are required to keep the gel together, and if you mutate those, the gel no longer forms. So then we can put a drop of this gel on a cover slip and wait until it's formed a gel, and then we can base it in the buffer. And I want to make the point that the denaturant are removed during the addition of the buffer, so there's no denaturant present anymore. And then we can also add fluorescent proteins and then ask whether the fluorescent proteins would accumulate in the gel. So we're basically imaging across the boundary between the buffer and the gel. And this is what we get. So if you look here at over time, we get more and more accumulation of PEX5 fused to GFP in the gel. Here's a quantification. If you just use GFP, there's no accumulation. However, if we use the N-terminus of PEX5, this is the unstructured region, and fuse it to GFP, the accumulation is just as good as with the full-length protein. On the other hand, if we just take the TPR domain, then there's very little accumulation of the protein in the gel. And importantly, if we mutate all the aromatic motifs, these WXXXFY motifs, then there's no accumulation of the PEX5 protein in the gel. And importantly, we can drag bound cargo into the gel. So here we're using unlabeled PEX5, and instead the cargo is labeled, so it's GFP appended to SKL, the PTS1 signal. As you can see, we get accumulation of the substrate in the gel. The accumulation here is not as good as before, and this is because this interaction is actually relatively weak. If we just use GFP SKL, there's no accumulation. If we mutate all the aromatic motifs, no accumulation. If we use GFP without SKL, no accumulation. But if we use PEX5, the N-terminal region, and fuse it to an antibody directed to GFP, we can drag GFP into the gel. And in this case, the accumulation is actually very good because this interaction is very strong. So these experiments lead us to this model where the PEX5 partitions into the YG phase inside the membrane. We believe that the YY interactions are replaced by Y interactions with the aromatic residues in these motifs that I mentioned in the N-terminal region of PEX5. And this partitioning would allow the receptor to move back and forth through the gel and take the folded cargo with them. So this is the model here. So we think that you form this complex, then PEX5 partitions into the 5G phase, can move back and forth, and eventually is moved all the way into the lumen. I'll come back to what the driving force for moving into the lumen might be. Okay, so then you have the receptor and the cargo in the lumen. The cargo is where it needs to be, but the receptor needs to return back into the cytosol. So how does this work? So we used a novel in vitro system that's based on Xenopus egg extract, and we identified an intermediate in the retrotranslocation of the receptor. And so this intermediate had about 30 amino acids or so already emerged on the cytosolic site, so we could cleave off these 30 amino acids by proteinase K. And then we used this intermediate, we pulled on the intermediate in a pull-down experiment, and then found that PEX5 interacts with a ubiquitin ligase complex that contains three components, PEX2, 10, and 12. So this suggested that the this might be forming the retrotranslocation. We also found the region in PEX5 that targets the protein to the retrotranslocation. It turns out to be an amphipathic helix that is also present in this unstructured N-terminal region. And then we determined the cryo-EM structure of this ubiquitin ligase complex, which is shown here. So PEX2 is shown in yellow, PEX10 in blue, PEX12 in red. We used an FAB fragment for better alignment of the particles in the cryo-EM images. And this was a collaboration with Satchel Aramilli and Tony Kosikov at University of Chicago. What you can see here is that there's a membrane-embedded region, and then there's a cytosolic region, and that contains the ring fingers. All three proteins are ring finger ligases. So ring finger 2, 10, and 12, they're forming what we call the ring finger tower. But more importantly, inside the membrane, if you turn it around and take away the ring fingers, you can appreciate that there is a hole in the membrane. And this is the pore through which proteins, the receptors, move back into the cytosol. So it's shown here. So the N-terminus of the receptors insert into the channel, and this allows this conserved cysteine that I mentioned at the beginning to be monoubiquitinated by one of the ring fingers. We think it's ring finger 2 that does the monoubiquitination. And then there's a AAA ATPase in the cytosol consisting of PEX1 and 6. This is a hexamer. It's a double ring ATPase. And in the ring, there's alternating PEX1, 6, 1, 6, 1, 6. And so it looks very much similar to CDC48 or P97. So this ATPase is believed to pull the monoubiquitinated receptor out of the organelle and return it to the cytosol. Now, as you can appreciate, that the hole in the membrane is way too small to allow this TPR domain to move through. So during retrotranslocation, when the ATPase pulls, the TPR domain is unfolded, and so it has to let go of the SKL sequence that it binds to. So this releases the cargo inside the lumen while the receptor returns to the cytosol. So this is what I said. So this is summarizing what I said about PTS1 import. So again, we're starting with this complex in the cytosol. It partitions into the YG phase here. It can move back and forth. There's no directionality of transport, like in the nuclear transport case. You can move the receptors back and forth. However, we think that one mechanism by which directionality is determined is by this other membrane protein that is a part of the docking complex, PEX14. PEX14 has a luminal domain that binds very strongly to these aromatic motifs. It's also a tetramer, so there may be avidity effects that help with the interaction here. And by interacting on the luminous side, you bias the diffusion towards the inside of the organelle. This may not be the only mechanism by which directionality is achieved, but we believe this is one of them. Then once the receptor is in the lumen, then the receptor has to return, so it inserts its N-terminus in the retrotranslocation, the ligase complex. You get monoubiquitination, and then this AAA ATPase pulls the monoubiquitinated receptor back out of the membrane, out of the organelle. TPR domain gets unfolded, cargo is released, receptor refolds, you get deubiquitination, and you can start a new import cycle. I want to make the point here that the retrotranslocation process, the recycling of the receptor, is actually quite similar to the EROD process. So, as you remember, in both cases, we have a ubiquitin ligase complex that is forming the conduit for moving the polypeptide chain back into the cytosol. In the case of peroxisomes, it's monoubiquitination. The ATPase is called PEX16, but they are actually very related to one another. So here you have the CDC48, P97 ATPase. And by the way, I should make the point that Yi Hong was the first one to demonstrate that CDC48 plays a role in EROD when he was a postdoc. So Anyway, so in both cases, there's ubiquitin ligase. In both cases, ubiquitination. Here's poly, here's mono. And in both cases, a AAA ATPase is pulling. So for the rest of the talk, I will be talking about PTS2 proteins. So some proteins have an N-terminal signal sequence. So it's shown here. There's not a lot of proteins that have this signal, but it's different, and it's at the N-terminus. And they need an adapter protein that is called PEC7. So PEC7 and the receptor and the cargo form a heterotrimeric complex in the cytosol. And the question is, does this complex also move through the phase into the lumen? And in order to test this, we reconstituted PTS2 cargo entry into the YG phase, again using these hydrogels. So again, we have a gel droplet. We had labeled cargo in the absence or presence of PEC7 and receptor, and we observed the accumulation of the protein in the gel. And down here you see the experiment. So if we have all components present, PTS2 cargo gets inserted, accumulates in the gel, whereas if you leave out either PEC7 or receptor, that doesn't work. PTS1 import in the same experiment is imported regardless of whether you have whether you have PEC7 present or not. Of course, it depends on the receptor as shown on this side here. This is weaker than here because this cargo actually moves faster into the gel. So experiments that I don't have time to show you, but we've now believe that this cargo complex also partitions into the hydrogel, and this interaction, this partitioning requires the receptors, WXXXF motifs, as well as an interaction of PEC7. We think the receptor is mainly responsive for moving through the phase into the lumen, but there's also an interaction of PEC7 with the phase, which I will show you in a second. So this means that the cargo complex ends up on the luminous side, and now the receptor recycles as shown before through the retrotranslocation. And this allows the PEC7 and cargo, PTS2 cargo, to be stripped off. Again, the cargo is where it needs to be, but PEC7 needs to recycle back into the cytosol for the next round. And so the question is, how does PEC7 get back? And it's very obvious it cannot use the retrotranslocation, that the ligase complex. It's way too big to fit through the pore. There's no unstructured region that could insert into the retrotranslocation. There's no cysteine that could be monoubiquitinated. So it needs to do it in a different way. And it turns out it goes back the same way as it came in, namely through the YG phase. And for that, PEC7 needs to associate with the phase. And it turns out that there is a short motif in PEC13 in this region that precedes the YG domain that reads KPWE. So this is a conserved motif in all species in PEC13. I should also make the point that when we use the hydrogel, the hydrogel also contained this region, not just the YG domain. Okay, so this motif binds on the PEC7 protein. So PEC7 protein is forming a beta propeller structure, and the KPWE motif binds on one side of the beta propeller. This is the opposite side to where the cargo, PTS2 cargo, and the receptor bind. So they can all bind at the same time. And I should say that we've confirmed this by bead binding assays. So the question is then, does PEC7 on its own partition into the hydrogel using this interaction of KPWE motif? So first of all, we can delete the KPWE motif, and this does not affect the formation of the hydrogel, as you can see here. And now if you use the PEC7 as a labeled protein, PEC7 GFP, you can see that in the Y-type gel, it will accumulate in the gel. But if you delete the KPWE motif, that doesn't happen, whereas PEC5 doesn't really care whether you have the KPWE motif present, because, as you know, it interacts just with the YG phase. However, PEG7 can enter the gel lacking the KPWE motif when it's associated with receptor and cargo. So again, this is the experiment that I showed you before. If you delete the KPWE motif, PEG7 on its own cannot enter. But if you add PEG7 together with the receptor and the cargo, it will enter the gel. So what we believe is that import is mostly mediated by the interaction of the receptor with the YG phase through the WXXXFY motifs. There's, however, also an interaction of PEG7 with the phase, and actually this interaction is the reason why the import is slower of PTS2 cargo than of PTS1 cargo. And export of the receptor of PEG7 is driven by the interaction of PEG7 with the KPWE motif in the YG phase. Okay, so now we're at this point where PEG7 partitioned back into the YG phase, but it needs to move from the YG phase back into the cytosol. So how does this happen? And it turns out this is a function of a new protein called PEG39. This is the protein that extracts PEG7 from the YG phase and moves it back into the cytosol. So it turns out PEG39 is a very small protein, and it contains two conserved features, an amphipathic helix and a motif that in yeast reads RPWE. So obviously it's very similar to the KPWE motif that is present in PEG13. So this is an AlphaFold prediction of how the PEG39 would bind to PEG7. And I should say this is also confirmed by bead binding assays. So as expected, the RPWE motif binds to the same place as the KPWE motif of PEG13. However, this amphipathic helix binds on the other side where the receptors bind and the PTS2 cargo binds. So, in fact, this helix here would allow the PTS2 cargo to bind, but it doesn't allow the receptor to bind. And I should also mention that although this interaction had not been seen before, but if you look at the conservation, there's all conservation residues on this side of the membrane, along the groove on this side and on this side as well. Okay, so now the question is PEG39 indeed extracting PEG7 from the YG hydrogels? And the experiment we do is to take a gel droplet, add PEG7 GFP so it accumulates inside the gel, and then we add PEG39 and ask whether it will induce the exit of PEG7 from the gel. And indeed it does. So here you see in a concentration-dependent and time-dependent manner, you can extract PEG7 GFP from the gel. And if you mutate, if you delete the RPWE motif, that doesn't happen, whereas the removal of the APH domain has only a very small effect. You might expect that PEG39 should not extract the trimeric complex on the way in, otherwise it would inhibit the import of the cargo. And indeed, if you form a complex of PEG7 together with cargo and receptor, the PEG39 has only a relatively small effect. So the next question is, how do you convert? So now you're at the stage where you have extracted PEG39 together with PEG7 into the cytosol, but now you have to recharge it with a new receptor and a new cargo. And this happens in 3 steps. And again, I'm not showing you the details. We confirmed this with bead binding assays, so I'll show you the AlphaFold predictions here. The first step, so this is the stage that was extracted from the membrane. The first step is that cargo binds. I mentioned that the cargo can bind despite the fact that the amphipathic helix is on this side as well. And in fact, it turns out that the amphipathic helix helps cargo binding. So there's a small but still significant effect on PTS1, PTS2 cargo binding to this complex. The next step is the receptor binding, and this displaces the amphipathic helix from the complex. So it's shown here. So now, the only interaction that is left is this KPWE motif of PEC39 binding on this side of the receptor of the PEC7 protein. And that is released when the complex rebinds to the face, because then the KPWE motif of the PEC13 displaces PEC39. So PEC39 stays behind in the cytosol. I should say that it also doesn't enter the hydrogels, and it's a cytosolic protein always. So this allows PEC39 to be stripped off, and you start a new cycle. So this is then the summary of what I told you. Again, I don't have to go through all the details here. So you have this trimeric complex, it partitions, it goes all the way into the lumen. The receptor returns to the retrotranslogon, stripping off PEC7 and PTS2 cargo. PEC7 reinserts into the hydrogel, and then it moves through the hydrogel on the cytoplasmic side. PEC39 extracts it into the cytosol, and then orchestrates the rebinding of a new receptor and ligand protein to start a new cycle. So, as you can see, PEC7 resembles nuclear transport receptors even more than the PTS1 receptors, because the nuclear transport receptors also go back and forth through the same pathway. And here we have a similar situation. And actually, the similarity is even more striking. There's a nuclear transport receptor called RAY1 or GLE2 in yeast that transports RNA through the nuclear pore. And it has a beta propeller structure, which is very similar to PEC7. And this one also doesn't interact with the EFG repeats of the nucleoporin, but rather to a short motif called the GLEPS motif. So it's quite similar to the PEC7 situation, where you also have where it also binds to KPWE motif, not to the actual phase-forming region. So finally, we think that PTS2 import might have evolved at the same time as nuclear transport because of these similarities. And presumably, PTS2 import existed before PTS1 import in evolution. And the reason we think this is because today there are only a few PTS2 proteins in most organisms. So, for example, only 5 in humans. And also some organisms like C. elegans lack the pathway altogether. So presumably it was replaced at some point by the more efficient PTS1 import pathway. And so this is my, I think, almost last slide. So what I told you in this talk is that we have 4 mechanisms by which proteins can cross membranes. The first one is the translocation through an aqueous pore. As I mentioned, we think this is a gated channel in which the substrate opens the channel, the plaque moves away, and you move in an aqueous environment. Then by studying ERAD, we found that there's a locally thin bilayer mechanism where these 2 proteins locally thin the membrane and allow the polypeptide chain to cross the membrane. And then we have 2 more mechanisms in peroxisomes. One, which is reminiscent of nuclear transport, movement through the YG phase, and for retrotranslocation of the receptors, we have a constitutively open pore, which allows the receptors to move back into the cytosol. And finally, I want to make the point that the last 2 mechanisms that I mentioned wouldn't work in other membranes, because peroxisomes are unique in that they allow the passage of small molecules. It's believed that molecules smaller than 800 Daltons can move freely through the peroxisomal membrane. Obviously, most other membranes are tight for even small molecules, so these mechanisms would not work in those organisms. in those membranes. So this shows the people who did actually the work. There are a lot of people who were involved in protein translocation into the R. This goes back 40 years or so. So I'm not listing everybody here, but these are the major people involved. Again, EROD was also many people involved. Here's Yihong, made a major discovery here. But there are several other people who are involved. I mentioned particularly work of Chudong, who solved the cryo-EM structure. And the peroxisomal protein import project was done by 4 people. I would say most of the work that I presented was done by Mike Scavura, who is just about to start his own lab at UT Southwestern. He did all the experiments on the hydrogels. He did all the experiments with the xenopus egg extracts. Pichang was involved in the structure of the cryo-EM structure of the retrotranslogon. He got help from Shudong, and also, as I mentioned, we had a collaboration with Tony Kosakov's group. And Yuan Ga did all the experiments on the in vivo system with the on the PEX-13. And I thank you for the attention.
Thank you. Please use the mics for questions. We don't have any online yet, but let me repeat the CME code. It is 57672. And let's refreshingly go to this side first.
Beautiful talk, and especially the peroxisomal import. But I have a question there, which is you nicely showed that PEX5 in vitro goes into the hydrogel. So I'm just wondering what keeps PEX5 in the cytosol in the cell, because if it was continually translocating into the peroxisome without cargo, that would be very energetically costly.
Right. So the question is whether PEX5 actually cycles with or without cargo equally well. We don't know the answer. It sounds like an experiment that we could do easily. It's not as easy as one might think, because some cargos can bind to other regions. So, for example, if you mutate the TPR motif so you don't have cargo binding, there are other regions that can bind cargo. So it's not easy to do. It's a good question.
Hi, Tom. Very nice talk. I had 2 questions. So in the PTS2 system, presumably the energy for the whole cycle is it all provided by the retrotranslocation of PEX5?
Yes.
And so even that helps with the gradient of PEX7? How does that work?
Yes. Yes, you're absolutely right. So the energy, I should have pointed this out, it's a very strange system. The import itself is ADP independent. The energy comes all from on the back end from the export of the PEX5 protein. So you need energy for the ubiquitination, and you need energy by the ATPase. And as you say, this energy drives both PTS1 and PTS2 import.
And then the second question, you talked about this PEX13 being in the membrane in 2 orientations, which is completely against everything you taught us in cell biology class. So how does that, basically if you have a hydrophobic sequence, it would immediately go through the side channel, but then why would it not do that, and how can it be in the opposite?
Right. So this brings up a very fundamental question that I think is still not answered. How are membrane proteins integrated in the peroxisomal membrane? We have some very preliminary data, and I hope nobody quotes me on that, because we don't really know whether it's really true. But we think that PEX13 might be inserted into the bilayer spontaneously, like a toxin. And presumably it does it both in the peroxisomal membrane and the ER membrane and any other membrane. The other membrane proteins, we believe, are actually all inserted into the ER membrane and then move from there to peroxisomal membrane by vesicular transport.
So you would have in both orientations depending on which...
And then the insertion, the spontaneous insertion of PEX13 presumably is a concerted process that will allow this dual topology to establish. It's like in the case of the toxins. We don't really understand how the alpha toxins are the ones that I'm talking about. They also insert into the pore, form a pore, and that also happens spontaneously.
Thanks.
Tom, on this side.
On the other side.
So, first of all, absolutely beautiful collection of mechanisms. I am very much intrigued by this local thinning mechanism. And I'm curious, are there some special lipids selected to be in this part? So are lipids a kind of cofactors there?
Well, we don't know the answer to this. So obviously the structures are way too low resolution to know where the lipids are. So, so we haven't seen, we've been able to reconstitute parts of this retrotranslocation with a reconstituted system with just HERD1. And in this case, we haven't seen a lipid dependence. But it's possible that the protein brought with it some lipid molecule step. And so I think the solution, the answer is still not really clear.
Thank you.
Yeah, Tom, that was a great talk. So my first question was already stolen, so I have to think of another one since I already stood up. So do you think that, what's the advantage of the HERD1, DER1 type system where you use membrane pinching to translocate what is probably a relatively hydrophilic polypeptide that we know can go through a hydrophilic channel?
That's a good question. I don't know what the advantage is. You know, curiously, this mechanism of local thinning seems to be more widespread than the actual aqueous channel model that we anticipated to be the more general mechanism. So it is, this local thinning you see with tail-anchored membrane protein integration. You see it with mitochondrial protein import. The twin arginine transport system in bacteria seems to use the same mechanism. So I don't know, maybe it's just a more primitive mechanism to allow translocation. My suspicion is it's slower than the translocation through an aqueous channel. But, it's hard to measure the rates, really.
Yeah, okay.
Yes, great talk. I also have two questions, and I'm sorry if I missed it, but once PEX5 is exported and it's ubiquitinated and unfolded, do you need chaperones to refold, and do you need a deubiquitinase to deubiquitinate?
Right. We haven't actually looked at this, so I suspect that the refolding may occur spontaneously, but I don't know for sure. It could be chaperone-mediated. It's likely that the refolding happens before the cleavage of the ubiquitin molecule, because there is some evidence, not from us, from another group, that ubiquitin is also unfolded during the retrotranslocation.
And my second question is because I was staring at the tyrosines, and they're different to the phenylalanine. Can the tyrosines be modified? Is that a way to regulate PEX5 and PEX13 phosphorylation, other the tyrosine modifications?
No, we don't think so. But let me make a point about the difference between FG and YG, because I think, some people might wonder, why is it that a nuclear transport receptor doesn't go into peroxisomes or vice versa? Why does PEX5 not go into the nucleus? And the answer is probably that the nuclear transport receptors don't like YG. They prefer FG, and they're relatively big, and the pore is relatively small. So that explains why nuclear transport receptors don't go into peroxisomes. The other way around actually may happen. So Dirk Ehrlich did an experiment where he blocked nuclear export with leptomycin B and looked for all proteins that accumulate inside the nucleus. And surprisingly, one of the most abundant protein that accumulates is PEX5. So it is possible that PEX5 is actually always imported and needs to be actively re-exported all the time. There's also quite interesting differences between YG and FG in the sense that the hydrogels that we make have a very different property than the FG hydrogels. The FG hydrogels, as you might imagine, because it's hydrophobic interaction, they become more stable at higher temperature, whereas ours, they melt like gelatin. So you can heat it up, the hydrogel becomes fluid, you cool it down, it becomes a hydrogel again, you can repeat this multiple times. So it is there. I think there's hydrogen bonding in the case of Y, and that may explain the difference.
Thank you for a really elegant and inspirational talk. It's great to escape from just one protein, aqueous pore. So I then had a couple of comments. One is in our hands, it looks like the other systems also, like the influenza fusion peptide, we're seeing a local thinning of the membrane as being key to pore formation there. And it's another system that oligomerizes. But the other thing I wanted to comment on was your answer about the peroxisome being leaky to molecules less than 800. And also, it reminded me of this Palladian dogma that membranes come from membranes. Except, I think the peroxisome, if I remember, is an exception to that, in that you can get induction of peroxisomes where there's apparently no peroxisomal membranes to begin with. So I think your mechanism of being able to find lipids and create an organelle makes a lot of sense in that regard.
Well, Josh, I don't think that conclusion is correct. So Palladio is still right. Membranes only are generated from membranes, and that's true for peroxisomes as well. So the membrane, the question is where do the lipids come from in peroxisome case? And as I mentioned, we think that there is vesicular transport from the ER. There may also be other non-vesicular transport of lipids from the ER into peroxisomes. So that's where the lipid comes from. You cannot form, people say that if you, for example, there are 3 proteins that have been implicated in peroxisomal membrane protein integration: PEX3, PEX19, and PEX16. PEX3 is a single-spanning membrane protein, PEX19 is a soluble protein, and PEX16 is not conserved. None of these proteins make any sense in terms of membrane protein integration. We think they are involved in the vesicular transport system. So basically, I think, there's no violation of the original dogma that George Palladi had. It's basically still Virchow's omnicellular, acellular. On the subcellular level, membranes can only be generated from membranes. Sec61 can only be generated from Sec61, and so on, right? So it's still the same.
I'll have to
We're not that revolutionary, I think.
I'll have to find the experiments that made me think this. Thank you.